Hi everyone! Today I'm going to talk about how we can use constraint programming to design microplate layouts. I'm currently part of a bioinformatics lab, which focuses on using artificial intelligence to develop systems that can aid with drug discovery. Laboratory work nowadays is mainly human-guided. Scientists use their knowledge and intuition to plan the experiments and decide what to try next. We would like to automate part of that knowledge so that a computer-guided system can help us with predicting safety concerns, explain drug mechanisms, screen for new drugs, and many other things. As you can see here, we currently have the equipment, but how do we go about designing the software that will run this automated lab? Well, one small but key component is deciding where to put each particular compound or drug and at which concentration in a microplate. In an ideal world, this will have almost no impact at all. All wells will be exactly the same, and we could simply place components in a pretty and visually convenient way like we have here in this picture, right? We have all the components lying next to each other, perfectly ordered. We have replicas of the same component and the experiments one next to each other, just to double check that the effect that we're seeing is actually happening, and it looks very nice and pretty. But reality is far from perfect. The outermost rows and columns suffer from something called edge effect, which means that the temperature and the humidity actually alters the experiment to the point of rendering those wells unusable. Instruments also happen to be imperfect. The pipetting robots can have problems with a particular pipe, and this can affect a whole row or column in every plate, depending on how your robot works. Even plate cleaning and handling is imperfect. Things like washing the plate once or twice can make a great difference between plates. Time makes a difference. Not everything is piped at the same time. We have an order, right? We piped either row by row or column by column or even well by well. And whatever you're placing first actually starts, happening earlier on. And this has an effect once you're trying to validate results. We have limited resources. We can't simply replicate all the experiments as many times as we would like. We don't have enough drugs for that. We don't have enough cells. We don't even have enough time. The experiments take 24 hours or 48. So we would run out of time and money. So how can we actually make things more reliable? And how can we get better results? Well, the scientists that has been working with this all these years actually have an idea of some constraints that will help making better and more reliable plates. So for instance, we can leave the outermost rows and columns empty. We can have replicas, and ideally we would like a whole replica, meaning a compound at all the different concentrations to appear in one plate. And this is so that we can actually compare in a way the effect of the concentrations between what happens on one plate. We would like then to make their replication on different plates. This is to compensate for whatever happens on this plate. Say, for instance, that it was cleaned a different number of times. We would like, if we have extra empty wells, to put them near the border. This is just because it's the most convenient place to put it, given that we know the rows, the outermost rows or column can have weird effects. Some people advocate for putting them just in the last plate so you can plan other experiments. We would also like to strike a balance between what is near the center and what happens near the borders. This is because, on one hand, the results that happen near the center are in a way more reliable. On the other hand, we know that there are certain effects that cluster depending on the location on the well. So we would like to be able to account for those effects and distribute them across all the experiments. So the real plate layout design problem would actually look something like this, where we have empty wells in the border and we have a whole mess of controls and compounds and different concentrations that are possibly difficult to do by hand, and those are also difficult to interpret by hand, which is why many scientists try to avoid this. On the other hand, if we have a robot which can actually do this, then we can take pictures and we can organize all of this by computer, and we can actually reduce the number of experiments that we need to do. So the input parameters that we have for this problem are basically the numbers and types of controls. Each experiment needs different types of positive, negative, blanks, and so on controls, and different amounts of controls that we want to place around. We also have a number of compounds that we want to test and a number of concentrations in which we want to test them. We have different plate sizes. The one robot is probably going to work with only one kind of plate, but in general, we want to be more general. And we have also a number of replicas, so how many times we want to run each experiment. And well, we could have other inputs depending on what kind of problem we have or alternative to the problem. So the variables are quite simple. We basically need to account for what kind of experiment or control happens at each well, and we just have matrices that represent the plates. We also have redundant variables here in which we check on which plate is located each experiment, and basically we do this to model more easily some of the constraints. Here we can see some parts of the mini-sync model that we implemented. We actually have a couple, 3-4 models in which we account for some of variants that we have talked about in the lab. But basically we have here a couple of constraints to account for the edge effect so that we leave the borders empty. And we have chosen to model this instead of just having a smaller matrix, because then this would allow us to also model the case in which we don't need to account for this effect. There are experiments that are actually more reliable and where we can use the borders. We are accounting, of course, that we have exactly the number of controls that we are placing, and that we have exactly one, that every experiment happens once and at most one. We're checking that all the concentrations per one replica happens in only one plate. And we finally have the channeling between the two models that I initially talked about, between having each experiment on each plate and then knowing in which plate is each experiment happening. We are far from done on this. We have, like I said, some mini-sync models that are running well at the moment, but we're still working on estimating the robustness of each particular plate design. We actually want to have a function that we can possibly optimize, or that can at least allow us to verify that this is indeed better, not only than putting things in order, but better than random. We're also working on including more alternative constraints based on which kind of experiment other labs are running. So these can actually be used for various labs and various kinds of multi-plate experiments. And we have a manuscript under preparation, so soon we'll actually publish that, hopefully, as well as the mini-sync model and some example data on our lab's GitHub. Well, I hope you enjoyed the presentation and thank you for listening. It's now time for some questions.